DNA replication! Woo! First, in DNA replication, you need your nucleotides. First, we have a DNA nucleotide. A DNA nucleotide has a phosphate group, a nitrogenous base, and a pentose sugar. It's pentose because it has 5 carbons. However, in a DNA nucleotide, this pentose sugar is known as deoxyribose. The base pairing in DNA nucleotides is different. G, or guanine, will bind with C, or cytosine. And A, adenine, will bind with T, thymine. And that's your base pairing for a DNA nucleotide. Next, RNA nucleotides.
The structure of RNA nucleotides is very similar to DNA. You still have your phosphate group, nitrogenous base, and pentose sugar, but this 5-carbon sugar in RNA is a ribose sugar as opposed to deoxyribose. The base pairing is also pretty similar, but in RNA, C pairs with G, cytosine pairs with guanine, and U, which is new to RNA, pairs with A, and this is because there's no thymine in RNA, so we use uracil.
Okay, so first, get out your DNA strands. So what you're going to do when you get these is the enzyme helicase will come and break apart the hydrogen bonds between these 2 parental strands and unwind them. This creates what's called a replication fork. Next, single-strand binding proteins will stabilize the unwound DNA to keep it from rebinding. The enzyme primase will come and begin to add RNA primer from the 3' end to the 5' end. Okay. As you can see, you can still see the base pairing. In DNA, the C and G will pair, and the T and A will pair, and so on and so forth. Next, the enzyme DNA polymerase III will come and begin to synthesize that leading strand where the RNA primer is already located, as helicase continues to unwind the strands. Again, base pairing continues and stays the same with DNA. As the leading strand is elongated continuously in this 5-to-3 direction, next the enzyme DNA polymerase I will come and replace the RNA nucleotides that were added by the RNA primer. However, as it continues adding, phosphodiester bonds will be created between the nucleotides on the phosphate sugar backbone. And that's your leading strand. Remember, it continues to add nucleotides at the 3' end and will never add them at the 5' end. Next, on to the lagging strand.
Now for the lagging strand. The lagging strand is formed pretty similarly to the leading strand. First, we need the enzyme helicase to come through and unwind the DNA molecule. This creates a replication fork. Next, single-stranded binding proteins need to come in and hold those 2 template strands of the DNA apart so that they don't reform into a double helix. Next, just like in the leading strand, the enzyme primase needs to come in and create an RNA primer. Since new nucleotides can only be added to the 3' end of the nucleotide before it, this is synthesized from the 3' end to the 5' end. Next, DNA polymerase III comes in and adds new DNA nucleotides one at a time to the 3' end of the nucleotide before it. Just like that. Now, as this is one fragment called an Okazaki fragment. So now, as helicase keeps moving down the DNA molecule, a new fragment needs to be created. These fragments are created because of the fact that new nucleotides can only be added to the 3' end of the nucleotide before it. Primase needs to come in and create a new RNA primer, 1 nucleotide at a time. Next, DNA polymerase III comes in and adds those new DNA nucleotides one at a time to the 3' end of the nucleotide before it. And once this is completed, that is our second Okazaki fragment. So we have here 2 Okazaki fragments composed of 1 primer fragment and 1 DNA fragment. So now we have to get rid of those RNA primer fragments. So we use DNA polymerase 1 for that, and that replaces those RNA nucleotides one at a time with their complementary DNA ones. Now here we still have those 2 Okazaki fragments, so we need a new enzyme called ligase to come in and create phosphodiester bonds between the sugar phosphate backbones of those 2 fragments. So now we have a complete new daughter strand on the lagging strand.